Hello. Can anyone hear me?
Yes, we can hear you.
Okay.
Great.
Okay. So you can also see me now, right?
Right.
Okay. So are we waiting a little bit or shall we start?
No, just 2 minutes, we will start.
Okay. I can also see Rakesh Tigade and Akansha Jain.
Yes.
So they are my college seniors actually.
So are you able to recognize them as well or not?
Yes, of course. Why not? But are you able to recognize me?
Yeah, I recognize you after I got the brochure. And
Okay.
after I saw your photograph actually.
Okay.
Hello, Kishan. How are you?
Hello, am I audible?
Yes, you are.
Yes, I am good. How about you?
Yeah, I'm good. Thank you for joining.
Yeah, Dr. Rakesh, how are you?
Oh, yes, I am there. Dr. Rakesh probably join in some time. And
Okay.
Dr. Akanksha Jain.
Hello, sir.
So yes, as you can see, we are 3 associated with the Department of Pharmaceutics. So now I think we will start. Dr. Rakesh will join in between.
Yeah, nice to meet you.
Thank you, Vishnu. Ajay, इधर आ जाइए. Okay, so good afternoon all. Myself, Dheeraj Beniwal, Dr. Rakesh Tikade and Dr. Akanksha Jain. On behalf of NIPER, we welcome you all for this webinar, Biosimilars, Global Scenario and Formulation Aspects. So before we begin this webinar, we must pay tribute to our national anthem, and therefore I request all of you to please stand up and give a tribute to our national anthem by singing the national anthem. Thank you all. Okay. The objective of this webinar is probably we all, will agree that, in 1990, the Indian pharmaceutical industry has started the journey of developing the generic molecules for the small molecules. And that is the reason that if you look at, the pricing, probably the medicines available at the India is at a very, very reasonable rate in comparison to all other countries. Not only that, India, the export of the medicine from the India to the other countries has also helped the nation in generating the revenues as well as, fulfilling the need. This is the reason that, India has played a crucial role in the healthcare. Now, we are moving into a new horizon. We can say that, now the therapy is shifting from the small molecule to the biosimilars or the large molecule. And it is essential that now we should also start our focus, again to move to the same journey that, we should also now come up with the, I should not use the word biogenerics. However, people use this, the biosimilars, because again, we all, acknowledge that biosimilar products or the reference listed biological products are extremely expensive, and therefore it is vital to have a biosimilars which are available or which can be available at a reasonable price similar to the small molecule generics. With this objective, we have started this, the series of webinars on the biosimilar development. In that, the first step is this webinar. So today we will have two presentations, one related to the formulation aspects of the biosimilar, and another is once we have any formulation ready, or in fact, before we start that, it is essential that we understand the market perspective of that product. And in that context, the biosimilar global perspective. So considering the important segments of this biosimilar, we have, organized this webinar. So moving to the first webinar, that is biosimilar development formulation aspect. Now, I would like to introduce our first speaker. So we have a Dr. Roshan Goswami. Dr. Roshan Goswami is a General Manager, mRNA Vaccine Division, Biological E Limited. He holds more than 10 years experience in various biopharmaceutical industries, including Abraxis, Spain, GSK, Italy, Novartis, Italy, and it is an honor that he also, got Madam Marie Curie Early Stage Researcher, the fellowship. In addition to that, he also, associated with the Santa Biotech, a Sanofi company. So with this brief introduction, now I would like to call Dr. Roshan Goswami for a talk on formulation aspects of the biological products. Dr. Roshan, over to you.
So hi everybody, and thanks for the nice introduction. Okay, so let me open my PPT. Share. I think now you guys are able to see something, right, on the screen, right?
Yes, we can see.
Okay. So basically, I am here, this Roshan Goswami, and as Dr. Beniwal has said that, yes, I'm here to talk about the formulation development considerations for biologics, or in India, you may more appropriately say it as biosimilars. Okay. So I have just put a caution slide here that please do not click pictures of PPT for publication and distribution purposes, especially for social media platform. Okay. I mean, this is just a caution because there is a long approval process for presenting anything outside the company and all, right? Although this is a very general thing and nothing much associated with company secrets, what I'm going to present here, but still keep it into consideration. Okay. So, I mean, moving ahead, my agenda would be who am I, which is already introduced by Dr. Beniwal. Why I'm here, a little bit about vaccine formulation, MAPS formulation, mRNA formulation, formulation characterization, and quality by design. Okay. So basically, who am I is already told by Dr. Beniwal, but I'll just give a little bit of brief that I'm born and brought up in Sagar. Okay. I don't know if I can use my pointer here. Can I?
Yes.
For recording, so start focus.
Yes, doctor, you can right click and then there is option for
Okay.
pointer.
Okay. Pointer option, yes.
Yes.
Okay, yes. Okay, so basically I'm like born and brought up in Sagar, Madhya Pradesh. So the other organizers are also from the same place. So hi everyone there. And so it's like almost 25, 26 years. I have never been outside my native place, and then it was all circuit of movements, right? So then I moved to Hyderabad in Sanofi doing vaccine formulation. Then I moved to Italy, where I got this Marie Curie fellowship, and I was in Novartis and GSK. It was the same site, but GSK took the Novartis vaccine division, and they acquired it. So I got opportunity to work in both the kind of companies, and they are really quite good, and I got a very good exposure there. and they really host a huge number of PhD and postdoc students. So like when I was there were almost 30 PhD students in the company and almost 50 to 60 postdoc students. So that is a good place to pursue your higher studies as well. Okay. Then from there, I was a little bit like unemployed, because after PhD, there is always this gap. And I was unemployed for almost 1.5 years. I came back to India, but nobody was giving me the thing which I needed. Nobody was identifying all these achievements. So 1.5 years I was jobless. Then I got an opportunity in Madrid, Spain, Map Science, where I was doing biologics formulation, and I was the lab head there. Okay, so that was a quite enriching experience as well. And during that time itself, Corona came, and then there was a huge thirst for Corona vaccines. And mRNA, on which I worked during my PhD in Italy, it gained a sudden importance. Okay, so suddenly there were a huge demand for people who has worked for mRNA, because many companies now want to put their leg into it. And that's how I moved back to India in Hyderabad in Biologically, who is willing to establish the platform of mRNA vaccines. And we have also got this WHO mRNA Hub COVID vaccine mRNA tech transfer, okay, which I'll be leading. Okay. So, I mean, maybe this is too much in a 30-minute talk. So let's move forward of why I am here. So like everybody here is related to pharma as a student or as a teacher or as an industry representative. So everybody knows that there is a huge round of evolution since the time immemorial, whether it is Ayurvedic or the vaccines, then antibiotics, then proteins. So like several things came. But since my core expertise is in formulation of biosimilars of different kinds, so I'm here discussing the formulation development and characterization requirements for vaccines, which are traditional vaccines and mRNA vaccines, and also monoclonal antibodies. Okay, so this will be the focus area. So, I mean, having said that, I mean, 30 minutes or 45 minutes is a very, very short time to discuss in detail all these things. Okay, so I did not put many things on the slides. I have just put it indicatively. More I would discuss the things. Okay, so what I am discussing may not be visible on the slide, but I'll try to make a story out of it. Some of the part is visible on the slides. Okay, so sorry on my part, I could not make a very thorough slide deck. Okay, considering the time frame, I'll try to limit myself to the next 30 minutes. Okay, so when it comes to vaccines, then basically before thinking about formulating a vaccine, we should understand what a vaccine is, okay, what it is made up of. So there is a long history of vaccine, but somewhere around 100 years back, a normal vaccinology has started, okay. Although if we talk about history, India and China are the main innovators in the field. Although because of all the colonialism and all these things, our contribution is not visible. So like talking about this Jenner, and after all these big scientists from Britain and all over Europe came up, the vaccines that we see today are wholesale vaccines, okay, which are basically some damaged bacteria, in simple terms we can say, which is not having virulating characteristics, but it can propagate itself and it can present itself to our immune system. Similarly, live attenuated viruses like MMR vaccine, measles, mumps, rubella, and there are several other viral vaccines of this kind. In fact, the recent vaccine of COVID, which is made by Bharat Biotech, is also something similar. Then genetically engineered and attenuated vaccines, in which instead of chemically attenuating them, they know the gene markers where attenuation should be done, and they do it through genetic modification. Then apart from it, so these are something which are like whole kind of vaccines, okay. So they are having multiple components of the pathogen to represent to our immune system. Then there was a trend of identifying the epitope, which is basically responsible for generating efficacy against vaccine. Okay, so which is basically saying that now we are going towards more purified forms, because wild strains are having a tendency to revert themselves into wild strains. Okay, all these inactivated viruses and whole cells, there are cases that have been seen that they have reverted back to the wild strains. Okay, like recently with oral polio vaccines. So that's why there's a trend of inactivated polio, which is genetically engineered. And there is also genetically engineered novel oral polio vaccine. Okay, so there are many trends coming out here. Then there was a trend. So similarly, what they have done is, instead of whole cell pertussis, there is acellular pertussis. So they are taking different parts of the bacterium, which they have identified as major epitopes, and then they are injecting, making a cocktail. Then there are polysaccharide and protein subunit. Protein is more stronger immunogenic than polysaccharide. So that's why polysaccharides like VIDT, like this called, typhoid. So typhoid is basically conjugated to toxoids like DT and TT toxoids to make it more immunogenic. So this is what I have shown here, that in traditional vaccines, as we go purer in vaccines, the safety is increased because the chances of converting to wild strain is diminished, but the efficacy is reduced. Okay. So whenever we are talking about whole cell and live attenuated virus vaccines, then there is no need of any adjuvant. Okay. But as we purify them more and more, their efficacy reduces with the increase in safety. So we need to make balance, and this is the reason why we use the adjuvants. Okay. So when we use all these adjuvants, so these adjuvants are basically nothing but like traditionally used one is alum-based adjuvants, and novel one is AS series adjuvants, Matrix M, QS21, CPG. There are different single-stranded, double-stranded DNAs and RNAs which can be used. And there are several compounds that are under consideration for novel adjuvants. Basically, they either trigger any immune pathway so that the vaccine become more immunogenic, or they try to present it in a better way. Okay, so the exact mechanism for especially traditional adjuvants is not yet known. Although for novel and adjuvant, there is a sense of mechanism. Okay. So basically, as I said, that as we purify more the antigen for which from which we prepare the vaccine, there is a reduction in efficacy and the need of adjuvant. However, there are also modern day vaccines. Although we should not consider protein subunit and polysaccharide vaccine and that they are very old, they are also new, and they are also first line approaches whenever any new pathogen or pandemic comes. Okay, so people try to prepare these vaccines first. Although now there's also a trend of VLP, which is virus-like particle vaccines, mRNA vaccine, the one that was introduced by Moderna and Pfizer, and viral vectored vaccines, which is sold by Serum for COVID, and also the Russian Sputnik vaccine. Okay, so these are also like a little bit newer platforms. So what in VLP, what do they do is they take the viral capsid of any virus devoid of any genomic material, and they try to put the antigen inside. Okay, they try to express it in a way that the antigen is already entrapped in the virus capsule, protein capsule of any specific virus. Okay, so this way, although it is purified form of vaccine, but it is more immunogenic and it doesn't need any adjuvant. mRNA vaccines, we all know it is a genetic material, although there are lesser chances of integrating it to the genome. So they are more safe, and they are basically the guides for producing protein in situ. Okay, so as soon as administered, they will express the antigen inside the body, and there is a specific formulation mechanism that we need for such vaccines. Okay, that we'll be discussing later. Then viral vectored vaccines like Chimpadino platform, AAV platform, there are different platforms. So there they just take a modified virus, which is not virulent, but which can present the expression of the protein. Okay, so they need to go to the nucleus. So mRNA need to reach to cytosol, and viral vector vaccines need to reach to the nucleus. Okay. So there are different challenges of preparing all such vaccines and all. So as I said, that VLP is designed to be more immunogenic, so it doesn't need any extra effort because by design itself it is more immunogenic. While vectors need to reach to nucleus, although these vectors, they are they are able to do it by themselves. Okay, so there is no need of any special approach because naturally viruses reach to the nucleus. Okay, so this is their natural approach. This is the reason why there is not much formulation development needed here as well. And mRNA, it is a different kind of setup that we'll be discussing later, and it needs lipid nanoparticles and all. Okay, so now let's discuss a little bit more in detail. Okay, so I cannot see the top. Just a second. Oh, yes. Okay, so formulating traditional antigen-based vaccines. Okay, so as I was describing in the previous slide, that what are the traditional vaccines. So, I mean, when we say that we want to formulate a traditional vaccine, then what comes into mind is either a whole cell, which doesn't need any specific adjuvant, then either live attenuated vaccines, which also doesn't need much of adjuvant. But when we talk about polysaccharides and the protein subunits, then we need a desirable formulation, which is with adjuvants, okay, to increase their efficacy. So what kind of interaction do we need with such adjuvants is that the antigen should be absorbed on the surface. Okay, so whenever we work with NDDS, novel drug delivery systems, we always try to encapsulate. Okay, but here we don't want to encapsulate. Here, our purpose is that the antigen should not be hidden from the system, but it should be exposed better into the system. Okay, so our strategy is to expose them better. Okay, on a bigger surface, so that they are recognized well by the antigen presenting cells. Okay. To do this kind of surface adsorption, what we need is interaction, okay, between the adjuvant and the antigen, and that is usually electrostatic attraction between them. Okay. So, okay, just let me show my pointer. So, if we want these two things to interact electrostatically with each other, then which attribute of the antigen becomes most prominent is the pi, or the isoelectric point of the protein. Okay, because for specific adjuvants, we need to put the protein at a specific charge for it to interact to the adjuvant. Okay, then I'll be discussing more about it a little bit later. Another important like general attributes is like physiological pH and isotonicity. Okay, so whatever vaccine we are giving, it should not be harmful immediately to the body at least. And to ensure this, we need to ensure the pH and isotonicity of the formulation, right? And then what is the desirable product presentation is that it should be all liquid product. It can be lyophilized as well, but lyophilizing with normal adjuvants is really difficult task. There is no such product in the market, okay? And people are still struggling to lyophilize such kind of adjuvanted vaccines. So having everything, all of these things into mind, what we have to basically do is that we need to ensure that antigen is absorbed into alum-based adjuvants. Alum-based adjuvants are the traditional adjuvants at pH 4. Okay, so this is the prerogative. This is where we want to work. So at pH 7.4, basically, we want that the antigen and the adjuvant should be oppositely charged. Okay, for it to interact with each other. So in traditional adjuvants, we have two options, which is like Algel, or which is like crystalline aluminum oxyhydride, which is not aluminum hydroxide, but is an isoform of that. Okay, so it is crystalline in nature, not amorphous. So when you think about lyophilizing it, this cannot be lyophilized in any case. Your protein antigen will be damaged immediately. Okay, So anyways, both are used in all liquid formulations. So although I have written here PI, which is isoelectric point, and since it is organic compound, there can be no isoelectric points, right? But this is actually point of zero charge, PZC. Okay, in simpler terms, I have written PI just for you guys to have a better memory of it. So the point of zero charge in this is 9 to 11. Okay, so whenever we put it in pH 7.4 or like pH 6 to 7, it becomes positively charged. Okay, I think you all are aware of the basic part of this PI. Why PI or PK is important is that if the pH is below it, okay, it becomes positively charged. And if we keep the pH above this point, then the molecule becomes negatively charged. Okay. So at 6 to 7 pH, this is positively charged. So whichever antigens are negatively charged, we will be formulating it with Algel. Okay. And another adjuvant is alphos. For it, the point of zero charge is 5.7. And it remains negatively charged because at pH 6 to 7, it will be negatively charged. It will be, the pH will be higher than the point of zero charge. Okay. So all the positively charged antigens can be absorbed into it. Okay. So this is like, seems to be a very simple formula here. Although we need to give, so whenever a drug substance is prepared, we need to give enough time for blending. It is not like immediate blending and immediate absorption. Okay. So we need to absorb it, then we need to keep it for maturation of this absorption process. So sometimes it is like 3 to 7 days job. Okay. So this is the reason why traditional vaccines takes a long time to make. Okay. In aluminum phosphate gel, there is another mechanism of binding, which is through exchange of phosphate groups. Okay. It is a very famous kind of interaction between hepatitis B antigen vaccine and this adjuvant. So the percentage binding is like 100%, okay, which is hardly reached in any kind of vaccine. So normally we target more than 90%, although in some cases it may be 60, 70%, 80% also, but 90 plus is the desired absorption rate, okay. Hepatitis B shows almost 100% with this adjuvant. So this is like the formulation of basic traditional aluminum hydroxide kind of things. Okay. What about the novel kind of adjuvants? Okay. So novel adjuvants, which are basically a specific path which activates the specific immunogenic path like CpG protein, MPL, single-stranded RNA, and I mean there are many more. If you will go into research, I mean it will be a very short time to discuss each and every one individually. Okay, but basically they activate a specific immunogenic pathway and they make the vaccine more immunogenic or more efficacious. Okay, so whenever they are like small molecules, then they are usually used along with alum-based adjuvants. Okay. And then a novel kind of adjuvant with a traditional adjuvant is formed. Okay. So like, for example, the Corbevax vaccine, which is released by Biologically very recently, like 3-4 months back, and which is like heavily used in childhood immunization for COVID. It is having the spike protein of COVID. It is having CpG adjuvant, which I have mentioned here, and alum base. Okay. So, I mean, this is the kind of formulation which it utilizes. And it is really safe because it is a protein subunit. It is not like Covaxin, which is attenuated or viral vectors approach. I mean, that is also less efficacious. So this is high efficacy and high safety vaccine. Okay. Then if we talk about other adjuvants, okay, so other than alum-based adjuvants, so they are also quite a lot. So there is also like emulsification system, MF59, okay, which is emulsion of squalene basically in some nano size, okay, like some 150 to 200 nanometers. This emulsion is formed. Although the mechanism of interaction and how does it actually work is not known. Maybe it is partly because of squalene, okay, because it is a precursor of several other molecules inside the body. So this is the reason why MF59 is used as an adjuvant, and it is used in flu vaccine for elderly, where it gives a strong immune response. Okay. Then there is AS series, which is by GSK. So ASO1 is a liposomal based, a liposome plus QS21 and MPL, okay, monophosphoryl So I mean, this is one kind of adjuvant system. Then we have ASO3, which is squalene, tocopherol, and surfactant, again an emulsion. This is another system. ASO4 is again, see, alum and MPL. What I'm saying here, that novel adjuvants like MPL will be used along with alum base. Okay, so they have just given a name as ASO4, but it is a very simple technique. In alum, they have added MPL, which is a specific immune pathway activator, and a novel adjuvant is formed. Okay, so this is the way all the traditional vaccines more or less are formed. Okay, so when it comes to characterization, let me go to the next slide. I don't know. Okay, so I'll talk a little bit of characterization here itself. Okay. So when it comes to characterization, basically, when antigen is formed, then it is in the form of either polysaccharide or protein or maybe mRNA or AAV. Okay, but when we are focusing about only this kind of stuff, which is traditional vaccines, then at the stage of DH, everything can be done. We can run HPLC. Okay, we know the native state of the protein. We know it is in a buffer. So we can check for aggregates, for the acidic basic species, and like whether it is aggregating or not. Okay, we can do whatever study we want. But once it is adsorbed, then our access to the physical and chemical nature of the molecule is lost. Okay. Then to study stability and all, we need to dissolve it. Okay, and when we have dissolved it, we have to dissolve it using harsh surfactants, or maybe acidic or base-based desorption. Okay, so we have to first dissolve it. And once we have dissolved it, then it doesn't remain in the same state as it was when it was absorbed. Okay, so it is really tricky to characterize all these kind of vaccines through normal physicochemical ways. Okay, that's why regulators want to see the immune response. Okay, so more about it, I'll talk later in the slides where I have specifically written something for characterization. Okay, so this is all about the traditional vaccines. Okay, now when we talk about mRNA, okay, so previously I was telling you that there is also VLP based, there is also AAV or vector based. So mRNA here basically like viral vectors, they can reach to the nucleus by themselves. You don't need to do anything for that. Okay, but the problem with viral vectors is that there is a chance of genomic integration, although slowly these guys will try to resolve it, okay, by making the viral vector more appropriate so that it doesn't integrate into genome. And there are already many gene therapy applications, like you have seen on, like you might have been seen on social media, Zolsengma, right? That, okay, this guy is dying, it is having SMA, it cannot move, and all these things. The therapy is really expensive, like 18 to 20 crores, okay, single injection. So that is the kind of thing that AAV vector therapy is able to do, okay. These platforms are able to treat undruggable targets, okay. So that is a very rare genetic disorder, and gene therapies are able to treat them. So that is a big feat for scientific and medical like field. So when we come to like LNP, okay, so another basic advantage that mRNA has is that like it has lesser feasibility to integrate with DNA because it is not a DNA, okay, it is an RNA at the end of the day. And usually for AAV vectors and other chimpadino or MVA vector, whichever vector you take, normally humans have some exposure of these viruses, okay. So their efficacy is quite low. So like I was stating this example of Zolsengma as a gene therapy. So whenever it has to be given to a patient, first of all, the sera of the patient is tested for the antibodies for the vector that they have used in the drug. If the patient is already having antibodies for the vector, then either it is giving a very high dose, so the cost of treatment increases further, or the patient is unable to respond to the treatment. Okay, so there are several risks associated. However, with mRNA, such risks are almost negligible. Okay, although our body never identifies, it identifies the foreign mRNA, and it tries to create antibodies against it. Okay, it tries to reject it because it does not accept it as our own body system, although the nucleotides are very few and they just rearrange among themselves to form different kind of stuff, right? Although our body is able to identify which mRNA is is of our body and which one is foreign. Okay, so this is the reason why we need to encapsulate it. Okay, because we don't want our body to identify the mRNA and start creating antibodies against it. So first of all, we want to protect it from immune recognition. This is the reason why we need to use a carrier. Another thing is it is extremely very sensitive for RNA's enzyme-based degradation. Okay, so if you have not protected it from mRNAs, then merely 5% of the dose or even less will reach to the cytosol. Okay, then forget about cytosol, it will reach towards the cell. Then you need to have the endosomal escape. Okay, because in endosomes the pH will be so low that mRNA will degrade. Okay, so if you are not putting it in any LNP or some kind of delivery system which is encapsulating it and providing this endosomal escape, then probably these kind of therapies won't work at all. Okay, so this is the reason why we have to work with systems like lipid nanoparticles, which is being used in Moderna and BioNTech vaccine, and other kind of gene therapies which are already approved with mRNA kind of agents, mRNA or other kind of RNAs. Okay, so basically it is composed of 4 parts, which is cholesterol, PEG lipid, cationic lipid, and I have written it's deuterionic, but basically it should be ionizable lipid. Okay, so we cannot give a charged lipid. So mRNA molecules are like negatively charged, and we want to give positive charged lipid here. But we positively charged lipids, when given as it is, they are very, very toxic. So this is the reason why we want to give something which is ionizable. By ionizable, I mean that its pKa should be somewhere around 6 to 6.5. Okay, so when we will be entrapping the mRNA inside the lipid nanoparticle, that we will carry out at a pH of below 6, so that the lipid carrier becomes positively charged and mRNA is anyways negatively charged, so they interact with each other electrostatically. And then we change the pH of the formulation before making the final drug product to somewhere around pH 7. So it goes above the pK of the lipid and it becomes uncharged. Okay, so this is basically the theory behind it. And usually there are many things which need to be considered. Okay, so cholesterol and DSPC, we know that they are structural lipids and all. PEG is important, although against PEG also body can generate antibodies. Okay, so our immune system may reject PEG chains, but still we cannot avoid PEG chains here because of manufacturability. Okay, otherwise we won't be able to filter them. It will clog the filters. Okay, so that's why PEG chain for manufacturability point of view is extremely very important. Okay, with other kind of drugs, whenever we talk about pegylation and all, we usually say that it rectifies or it passes through the long circulating kind of liposomes, right? But with research and all, depending upon the length of PEG, actually they can be detected and they can be cleared from the bloodstream even quicker. Okay, so there's a lot of research behind that, and people are trying to optimize their length and all to be used, and there are several patents for that. So usually, this is the hypothesis behind using all this. And then we have to optimize the N/P ratio, like phosphorus to nitrogen ratio, just to determine how much mRNA your system can entrap. Okay, The pH thing I have already explained that the pK of the lipid should be 6 to 6.5 so that we can formulate somewhere below that pH to make it positively charged and then bring back it to normal physiological pH. Okay. Then another thing is, I mean, these things I have already discussed, right? So basically, how do we make it? Like manufacturability is quite easy nowadays. Previously, there was a PNI system, a microfluidic based system, which was used, and they are having this system for multiple scales, for lab scale, pilot scale, GMP scale, and like all the Moderna and Pfizer vaccines are made using these systems. And now there are other players as well, which are bringing more efficient systems. But basically, it is using the ethanol dilution process. Okay, and in lab it can be even made through a handmade T-junction apparatus. Okay, it's quite simple for research purpose. So basically in process development, we have to optimize different flow rates, the composition of the lipids, the N/P ratio. Okay, so when we optimize all these things, then you have the product which you know how it will behave during the entire length of manufacturing and for many batches to come. Okay. So, I mean, this is the system which is basically used now, but what is the need of the day? Okay, I mean, this I have highlighted here. So basically, to be able to sell this kind of product, basically you should have a proprietary ionizable lipid. So the deuterionic lipid or the ionizable lipid that I was talking about. So these other three components, they are not patent protected. But this fourth component, which is the most important component, this is like patent protected whosoever is researching on it. Okay. So like Moderna's lipid is patented for themselves. Pfizer's lipid is protected for themselves. Even other companies who are trying to do so, if they, so I mean, other companies who are trying to do so, they have to procure another, they have to make licensing agreements and all with other lipid provider that will give a proprietary lipid to them. But still having a proprietary lipid also, Moderna is facing like 2 to 3 lawsuits against the delivery system itself. Okay, so although they have earned like 15 billion or so in a very short span of time, but they have to face a lot of lawsuits against these kinds, I mean, against using this kind of LNP system because it was first used by Alnylam, which is a Sanofi company. Okay, so they said that you have not given us credit and all these things, and now they are demanding money from them. So it's quite a risky business even today. So basically the need of the day is if you want to work with it, then you should develop a proprietary ionizable lipid. If you have done it, you can earn millions or maybe billions by selling it to some company. Okay. Another thing is thermostability. So as you have seen that this Moderna and Pfizer vaccines, they are cold storage, like -20 or maybe even lower. Okay. So what we need to work on, and there is a huge funding on developing thermostable vaccines, which are at least stable at our refrigerator, like 2 to 8 degrees for at least a few months. Okay. Because people who develop them, they know that it cannot be stabilized for much longer. So they are also not expecting a huge 2 to 8 storage time. Even a 3 to 4 or 5, 6 months is more than enough. Okay, so that at least transportability can be taken care of. And if it can be made thermostable for 2 to 3 years, then it is a big feat. Okay, for such kind of vaccines and therapeutics. Then another thing is like whenever you are improving the thermostability or bringing new ionizable lipids, you should have this balance, which is the balance between stability and efficacy. Okay, you should not be bringing something which is compromising the efficacy, because if you have followed the COVID vaccines clinical trials, then Moderna and Pfizer vaccines are like more than 95% efficacious. But this Oxford vaccine which Serum was selling was hardly 70% efficacious. Okay, and this for the reason that I told, that maybe there are already antibodies against the vector itself. Okay, so these are highly promising, very highly efficacious. So whatever experimentation we need to do, we have to be extremely very careful about it. Okay, I think I'm already towards the end and I still have to discuss many things. Okay, so I'll leave this topic here. I'll move on to the next. Okay, So about characterization, okay. So I was already telling that adsorbed vaccines need to be dissolved, okay. Then high order structure will not remain the same, okay. Same goes with mRNA because this need to be de-encapsulated, okay, before testing. So the final DP which is inside the liposome is not the same as what after de-encapsulation. So you need to be extremely careful. So especially for vaccines, what regulators say that you should prove the like efficacy in animals, okay, the immunogenicity, vaccine immunogenicity or potency. So you have to wait for 2 doses, which are at least 1 month apart, okay. So the quality control testing is very long for the vaccine, is like almost 1 to 2 months, okay. So this is the reason why mRNA is getting more attention because it doesn't need much to manufacture. It is very fast. Within a week time, you can prepare a batch, okay, and you can then, if you plan correctly, then you can release 1 batch per day. But with conventional vaccines, it will take at least 2 to 3 months, okay. So this is the reason why all the companies are going towards mRNA platform. Then moving next, I'll try to be a little bit quicker. Okay, I've tried to make it more interactive, but I'll try to make it faster now because time is a constraint. Okay. So, I mean, we have talked about different vaccines, but what about the route of delivery? Okay. So, I mean, usually I think everybody knows here that vaccines are traditionally given intramuscular. Okay. I would have asked why not IV and all these things, but we don't have enough time to discuss all these things. Okay. So basically, like conventional vaccines, they are given uh, IM route, and exploratory vaccine delivery route is intradermal. Okay, so in intradermal also it will not be given by injections, but it will be given either by patches or through a patch gun, okay, or a microneedle gun. So several microneedles will enter your skin, and this is under like high level of development. Okay, so these 2 therapies are going to come soon. Okay, this kind of delivery systems. Then why don't we give it IM? Which kind of things we give IM? Okay, so basically protein therapeutics is what we give IM and subcutaneous. Okay, IV route and subcutaneous route, these 2 players. So that's where we enter the discussion of MAPS formulation. Okay, although we are towards the limit, so I'm not discussing here too much. So usually MAPS are given by IV, but there is a huge trend towards getting it delivered through subcutaneous route. Okay, there are already several monoclonal antibodies in market which are delivering through subcutaneous route. So through IV, the concentration is up to 25 mg per ml, and through subcutaneous route, it should be 250 mg per ml. Okay, so the 2 biggest challenges are when we are doing something for the subcutaneous delivery, because now it is more preferred route. The biggest challenge is the viscosity, because when you concentrate up to this concentration, then basically it becomes a gel, and it is very hard to process, especially for intravenous or like injectable purposes. Okay, gel for therapeutic dermal application is fine, but when you are developing something for injecting, then it should be easy to prepare. Okay, because it needs to be filtered out. So basic biggest challenges is viscosity and stability. Okay, stability because all the proteins here, like all the proteins are having phenylalanine, tyrosine, and tryptophan, which are like more organic kind of amino acids, and they combine with each other, and basically they aggregate the molecules. Okay, and whenever any aggregated map goes into the bloodstream, it gives a huge immunogenic response, and people may also die. Okay, that is anaphylactic shock, what we call. So that is a very serious condition. So we need to solve these 2 challenges. Usually here also, now here we are not making it with any adjuvant. Okay, it is all liquid, and only some excipients and protein will be there in the formulation. But is PI of protein important when there is no physical interaction? This is the question that I wanted to ask, but I'll give the answer. Yes, because basically whenever any protein is in a buffer, then the point of 0 charge and the PI are quite different. Okay, so basically I will not go very deep into discussion, but they are usually, the thumb rule is that you should formulate it 4 to 5 pH down the pi of the protein. Okay, usually the pi of MAPS is 8 to 9, so you should be formulating it 5 to 6 pH. Okay, and then it is more stable and less immunogenic. Okay, then what kind of buffer should be used? So since I said that viscosity is a huge challenge, so the buffer should not be multivalent. Okay, they should be univalent as much as possible, like acetate. So like we should avoid completely citrate, phosphate kind of buffers, and we should be using more of histidine, acetic acid kind of buffers, which are univalent, and they are weaker buffers. In fact, there are a few formulations of innovators which are buffer-free formulations. Okay, so the stigma of physiological pH and physiological osmolality, like 360 or isotonicity, whatever you say, that is gone here. Okay, so even buffer-free formulations works perfectly fine, and they are even more stable and even more well tolerated than the other kind of formulation. And also here we give, like, either they can be presented in a vial or pre-filled syringe or a pan injector. Okay, so these all three things are available. This is not future, this is what is currently running. Okay, so I mean, we have to look on what are the features that we need to characterize for making all these things, all these three things as a final drug product, because everything has their own requirements to be tested. Okay. Then basically in characterization, you have to do structural determination through like CD. Okay. 2D, 3D structure can be ascertained. Thermodynamic properties, you can use any dye-based test by giving a heat ramp, or maybe a DLS, you can do a DLS on heat ramp, and micro DSC you can do. Then you need to ensure what are the stability indicating properties while doing the degradation studies. Okay, so you intentionally degrade your molecule by exposing it to different conditions, and you will know which conditions are more prone to degradation. And during formulation development, you may make use of this information to make a better formulation. Okay. Then you need to get rid of structural isoforms, but usually formulation people don't do it. Downstream people, purification will do it. Okay, they will get rid of the acidic basic species. Okay. Then general parameters, as I said, that also buffer-free formulation. So the stigma of pH and osmolality is gone here. So you can do it on very less osmolality formulations also. They are very well tolerable and very, very stable. Okay. Extractable leachables you need to study depending upon the kind of drug product you want to prepare. Okay. Then I will just give it a thought to you guys as photo stability studies. Okay. And in-use stability studies. So the students may look into these things. Okay. These are just keywords, but whenever you are appearing for any interview in industry for biologics formulation development, if you will talk about them, it will give a very good impression. Okay. Then last but not least, so what we have discussed is very basic thing, and this is like more elaborated thing. So down the line of all the process development from scratch to the final introduction to the market, we have to be very sure that our process is robust and all. So this is just a snapshot. I mean, this is a very detailed discussion in itself, but I'll just put a very, very brief view of that. So basically, whenever we are at, like, we have to understand the process, how we are going to formulate it, because usually after TFF, there is no blending step. So we have to do all the formulation development before the concentration of the protein itself. Okay. And during the protein concentration, excipient drift happens. Okay. So we have to have a very good process understanding. We have to have a very good critical quality attributes understanding, that which attributes will finally diminish the properties of the drug, or they may be like harmful for the patient. Okay. Then we have to start our risk assessment. FME approach is the most common approach. Okay. Where what we give is we risk rank according to a table like this. Okay. So we have severity, occurrence, and detection. So like how severe is the problem? Okay, like suppose you are considering pH. Your pH range should be 5.5 to 6.5. Okay, so what if your pH is 5? Okay, so what is the severity? What is the chance of happening it during the process? And are you able to detect it? Okay. So depending upon this, we make a risk assessment matrix. Okay. And we check all, so like if the severity score is 1, occurrence is 1, and detection is 1, then it is less severe. Okay. So we don't study it. Similarly, depending upon this risk rank scoring, whatever falls in red, you have to study them. Okay. And you have to study them considering like this kind of setup. Okay. And what is the edge of failure? What is the proven acceptable range? Then after your experimentation, what is the normal operating range? And what set point you have put across? Okay. What are the action limits? Okay. So, I mean, this you have to do for each and every single process parameters. Okay. And material attributes. Okay. So once you will do the risk assessment, you will score it. Then you will do a process characterization and you will define all these things. Then you will do a post characterization risk assessment, where maybe a lot of these red ones will go green or yellow. Okay. Then you will design a summary of your process, and then you will draft a control strategy, which will be taken up by manufacturing, and then they will decide, okay, this feasible, this feasible, this not feasible. Accordingly, they will take a control strategy to be implemented into the manufacturing process. Okay. So, I mean, I'm not wasting my time giving up the summary, but thank you guys. And I think this has enlightened you up a little bit on the formulation development. Think about it and like be innovators, bring something new to the industry. Okay, we'll be very happy. Okay, so that's all guys from my part.
Thank you, Dr. Akshan. It was very, very informative talk. Thank you so much. So we can have a couple of questions from the the audience, if any questions.
Yes, Dr. Roshan, this is Kishan Suchak. So it was a very nice session about vaccines, mRNA vaccines, as well as formulation aspects. I hope I'm audible well. Yes.
Yeah.
So just one thought on like what is your perspective on vaccine therapeutics? Because as of now, the world is looking for vaccine as a prophylactic.
Yes.
So now very few companies are preparing now for vaccine for therapeutics like onco segment and other therapeutics.
So in fact, basically the BioNTech company, which has introduced first almost the COVID vaccine, right? They are all into therapeutic vaccines only. Okay. So I know that company from like almost 10 to 12 years from now. They were having all the funding for cancer-based therapeutic vaccines. Okay.
Right.
And like industry is already working towards it a lot, and it will be not only therapeutic vaccines, but they will be personalized vaccines. Okay. So there are some like common mutations in the genome which can cause cancer. So for such kind of mutations, they can create a mass vaccination. Like HPV vaccine is also one of the prophylactic as well as therapeutic vaccine. Okay. Although it is a protein, but what these guys are doing in personalized space is that if you are having a cancer which is not a common mutation, then they will bring the patient, they will take the sera, okay, they will test the mutation, and then they will develop specific vaccine, a specific antigen against your specific mutation, and they will treat you. So the cost of such treatments will be very high, and people has to be having the health insurance for sure, a very good health insurance. Like for Zolsingma, Novartis has given time for health insurance guys to pay on installment.
Yeah.
Okay.
That's
so
correct.
they are such expensive medications that even health insurance guys are unable to pay it one shot. Okay. So I mean things are moving definitely, and it will like cost will also reduce a lot. But like you can see many therapies coming in some 5 to 10 years time, but its cost effectiveness and all you will see in 25 to 30 years time. Okay.
Yeah. Thanks.
Any other question from the audience? If not, we can move to our second speaker. Okay. Okay. Once again, thank you, Dr. Roshan, for a very, very nice talk. And now let's move to the second speaker. So now it's an honor for me to introduce our second speaker for the today's webinar. So we have Mr. Kisan Suchak. Mr. Kisan is a manager, business development, portfolio, and strategy at Casio Biosciences. He holds more than 8 years of experience in various pharmaceutical industries, including Amneal Pharmaceuticals, Alembic Pharmaceuticals. And with this, the brief introduction, I invite Mr. Kishan to deliver his talk on biosimilars global perspective. Mr. Kishan, over to you. Thank you.
Yes. So I hope my slides are visible now.
Yes, we can see.
So I'm making it slideshow first. Okay. Yeah, so first of all, thanks a lot for the organizers, NIPER-Ahmedabad and all the organizing team, including Dr. Dheeraj Rakesh and the Madam Akanksha. So yeah, it was a wonderful session from Dr. Roshan also. And now I'm moving towards biosimilars, which is I'm talking about more on global scenario. And while Dr. Roshan has focused more on vaccine and formulation aspects for vaccine as well as MAP, I'll be more talking about biosimilars, including USA, Europe, as well as a scenario in India. So yeah, thanks for the brief introduction, Dr. Dheeraj. But let me go through my detailed profile with education and working experience. So my education basically covers Bachelor of Pharmacy from LM College of Pharmacy. But then I completed my MS Pharm from the same institute, NIPER-Ahmedabad, from the pharmaceutics. Then with the job, I have completed my Indian patent agent as well as trademark agent exams. So I have cleared both of them, and then I have completed Bachelor of Law, that is LLB from Gujarat University. Then from the BK School of Management, I have completed International Business Development. And from the Harvard University, few courses I have completed with respect to finance and economics. So basically my profile is three dimension of techno, legal as well as commercial part. And then with working experience, I started my career basically from Alembic Pharma, but then I moved to Amneal Pharmaceutical, where I was looking after patents and IPR aspects as well as portfolio management like new product selection also. And then I joined Kashyap Bioscience, and recently I'm working here and looking after business development, portfolio and strategy role. So with this, let's start with the presentation. And as I have stated, like I'll more talking about biosimilars. So I'll covering biosimilars definition, regulatory agencies. What are the regulatory agencies across the globe? The key regulatory agencies. Then biosimilars versus generics. What are the key differences? Then biosimilar development and challenges associated with the global biosimilar business. Then I'll talk about global opportunities, like why companies are moving toward biosimilar business and what are the opportunities. Then we'll move towards a few territories, like we'll talking about biosimilars in USA, Europe, and Indian perspective. Then a few interesting topics about interchangeability in biosimilars and extrapolation of indications. And finally, as a summary, what are the key points to consider while going global? So starting with just a minute. Yeah, so starting with first topic, that is biosimilars definition and regulatory agencies. Just a minute, I'm changing my view so I can view it properly. Okay, So basically, we're talking about the USA regulatory agency, Europe and India. So in USA, it is US FDA, Center for Biologics Evolution and Research. So CBER is taking care of all the regulatory aspect with respect to biosimilars. In Europe, it's EMA. In India, we all know that it's SADESCO, Department of Biologics. Now, as you can see, in the definition of biosimilars, if you see in the USA, a biosimilar is a biological product that is highly similar. So unlike generic, it is not an exact copy of innovator product, but it is termed as a highly similar to and has no clinically meaningful differences from an existing FDA approved reference product. In the Europe, they term it like a biosimilar medicine is a medicine which is highly similar. Again, here it is a highly similar and not the exact copy, unlike generics, which is similar to another biological medicine already marketed in EU. So that is termed as a reference medicine. And then in India, biosimilars are termed as a similar biologic product. And the definition is like a similar biologic product is that which is similar in terms of quality, safety, efficacy to an approved reference biologic product based on comparability. And there is a key provision in the US and Europe that is called innovator product exclusivity. Like once the innovator product got approval, after how much time the biosimilar company can launch its product. So that is based on exclusivity. Patent part is totally different, but here we are talking about biologics innovator product exclusivity. So in USA, it is like there is a flat 12 years of total market protection. So once a biologic product. So when I say biologic product, it is called innovator product. And when I say biosimilar product, it is a similar product to biologic product. So in USA, it is 12 years of total marketing protection, which is 4 plus 8. So 4 years before an application for a biosimilar may be submitted. So you can submit a biosimilar application after 4 years of biologics approved, and you can launch it after 8 years. So total 12 years. So once 12 years has been completed, a biosimilar company, if the product has been approved, they can launch into the US market. In the Europe, the provision is for 10 years. But in India, as of now, it's a lack of market exclusivity provision, but we may see in the future such kind of provision may enter, and which would foster competition into the market. So amongst all these key regulatory authorities, USA, Europe, and India, Europe was first in the world to formulate the policy framework for the approval of biosimilar product. And in Europe, the first biosimilar approved in 2006, which was Omnitrope by Sandoz. So you can see the gap between first biosimilar approval in US and Europe. So in Europe, it was approved in 2006. In US, it was approved in 2015, which was again by Sandoz filgrastim product. Now talking about why there's a difference between biosimilar and generic. So these are the key differences wherein if we talk about on the left side generic medicine, on the right side it's a biosimilar medicine. So if you talk about generic medicine, those are small molecules. It's produced by chemical synthesis having less than 500 to 900 Dalton of molecular weight, wherein biosimilar product are obtained from the biological source having 140K around or more than 140K Dalton of uh, molecular weight, and hence since it is been obtained by biological source, the exact copy is not possible because of its unique manufacturing methods and its natural biological variability, where we can expect similar or same molecule in case of generic. Now if you talk about development perspective for generic, then it is mainly based on demonstration of bioequivalence. It is not required to demonstrate lengthy clinical trial or safety or efficacy studies. Very few, like complex generic, in case of complex generic, it may require we need to prove safety or efficacy, but otherwise it has been most of the case demonstration of bioequivalence. But in case of biosimilar, it is a demonstration of using comparability studies, chemical structure evaluation, highly similar biological function, efficacy, safety, immunogenicity, all these studies the biosimilar company need to perform. That's why it is getting more costlier development and also time consuming development. So as I told, like clinical study is not required for generic medicine, only peak pharmacokinetic bioequivalence studies would suffice in most of the cases. But in case of biosimilars, you need to perform comparable similar safety and efficacy trials. If you talk about cost of development for generic medicine, it is somewhere around 1 to 5 million USD or maybe lesser than that. But in case of biosimilar product, the cost is huge and it is coming around 40 to 80 USD. And in some case, it may go higher to that. So you can consider there is a significant difference in cost of development in generic as well as biosimilar. If we talk about price discount, like if the product is there in market, that's called a brand product. So brand product discount, price discount from brand product, it is more than 85% and it may go up to 95% in case of generic medicines. But in case of biosimilars, it is not that much huge price discount. So it is around 20 to 30% of price discount. And if you talk about substitution, that is interchangeability. So this is not like generic medicine where it is easily substitutable by pharmacy or it is easily switchable by physician. The biosimilar medicine are not easily switchable, or it is required to prove interchangeability or switching studies based on country policy. Not all countries are providing such designation, like they are not evaluating interchangeability. But yeah, in USA, it's a strict requirement, like to get designation for interchangeability study. They need to perform few trials for switching establishment. Yeah. So next is originator versus biosimilar development. Now, when I say originator, that's a brand product. So on the left side, this is a you can say pyramid of originator product, where the key requirement is clinical studies. So the pyramid is like the burden for analytical is lesser than then preclinical they need to prove PKPD, and then clinical profile. The pyramid is exactly reverse for biosimilar development. The key area for biosimilar company which need to establish similarity with the reference product is analytical characterization. And then based on the similarity studies, they need to go for preclinical, PKPD, and clinical studies. But why clinical part is lesser compared to originator? Because in case of biosimilar, the clinical trial which need to perform is on single indication. So the lengthy trials are not required. They just need to establish that their mechanism is working. And even if they prove on single indication, even if they prove efficacy on single indication, they can get indication based on extrapolation of indication. So that is why clinical part is lesser compared to originator. But yeah, this is need to be performed. And yeah, as I told, like because of higher molecular weight in case of biosimilar, the complexity to prove the similarity is getting difficult compared to generic medicine. And at the same time, analytical certainty is also increasing for uh, biosimilar products. So the key part is analytical characterization, and then followed by phase 1 and phase 3 studies. So I have summarized few challenges which are associated with biosimilar businesses. So when we are going global, these are the key challenges, or we need to consider this part. So the company need to consider cost of biosimilar development, which is ranging from 40 to 80 million, and can go beyond that based on which market we are targeting. So as many market we are targeting, we need to perform few studies according to their reference product. Then talking about resources, so most of the Indian companies have limited resources as compared to companies in the West. So here the limited resources, what I mean is not by manpower, but it is by uh, structure of their process department, as well as their reach towards market, their reach toward physicians. So those kind of resources are limited for Indian companies compared to West. Then if we are talking about process part, in biosimilar, key part is processes. So process are always patent protected by innovator companies, and any change in manufacturing process, such as use of different cell lines, growth medium, and protein purification processes, can result in variation in the final product. So if we change the processes, we may end up into the different product or variation in the product, which may be threatful to analytical characterization as well as safety, efficacy, immunogenicity. So all this can be compromised based on change in process. So that is one key challenge a biosimilar companies are facing. They need to enter to the market early, as well as they need to bypass the patent also. Then analytical characterization, as I mentioned, sophisticated techniques required, and which are the heart of overall development. Now talking about safety and efficacy, as I told, the biosimilar company has to at least one clinical trial they have to prove, and then they have to perform immunogenicity study also. So this is one key requirement and differentiated requirement compared to generic products. And if we talk about overall development timeline for the biosimilar product, it ranges between 6 to 10 years based on complexity of molecules. Then one key part after developing the product is its scale up and commercial manufacturing. So it requires a reproducibility of processes, robust process, and lower batch to batch variation, high percentage yield, so that the cost of development would reduce. Then if we go about marketing strategies, then it requires a well-structured and skilled set of professionals to run the patient's assistant program. It is not like simple generic medicine where it is directly therapeutically substitutable by a brand product. Here, the marketing would be like a brand product itself. So biosimilar marketing itself required a huge task force, and they need to educate physicians, healthcare providers also with respect to their product. Then last part, which is dynamics towards uptake and adoption. It is not like once you are into the market, your product will have good adoption or good uptake. It's so for that one has to consider one has to consider uh, individual countries reimbursement policies. So whichever countries are targeted for the marketing, their reimbursement policy, their pricing policy, then what are the factors like physicians confidence to switch patient to biosimilars. So all this market research one has to conduct, and then the decision to go into the market is been taken. Yeah, so now I'm talking about the opportunities, why the companies are more looking after biosimilars, and what are the opportunities. So if you can see in this graph, there is increasing USA biosimilar medicine approval. So it has close to 60 number of US FDA approval in 2016 to 2020 time period. So it is getting increased. So the more biologics product getting approved, like more innovator product getting approved, there is a huge opportunity for biosimilar also. Of course, time will patent protection would come into picture, but yeah, that is a huge opportunity till 2030. And if you see about total global biologic market size, it is close to 500 billion USD. So it is estimated that by 2026, this kind of growth would be there in total global biologic market. Same way in the Europe, you can see there is a growth in biologics market, and biologic represent at least around 34% of medicine spending in Europe, with 78.6 billion in 2021, and it is growing at a rate of 10.5 CAGR over the past 5 years. So this is showing like biology product are getting increasing the market size in Europe also. Now coming to our patent part. So you can see in this chart, like from 2012 to 2028, these are the key products for which patents are getting expired, and the total opportunity is more than 240 billion by 2030. So you can see like there is a huge opportunity, and that's why Indian companies are nowadays more looking towards developing these biosimilar products. Now we'll go towards USA territory, like what are the scenarios, what is biosimilar uptake and market share in USA, biosimilar price dynamics, reduction in total drug spend, and finally we'll talk about biosimilars projected saving in USA. So this is just a informative slide where in United States, total 36 number of biosimilar products are approved by US FDA, and because of patent hurdles or lawsuits, there are 21 products approved. So the first product which was approved in USA biosimilar is Zarzio, that is filgrastim, in 2015, back in 2015, and then total 36 number of biosimilars has been approved. Kashiv, where I am working right now, they have recently got approval for two products, that is filgrastim, and then recently in the last week, Kashiv has got approval for pegfilgrastim also. So these are just an informative slide where the scenario you can consider as a total number of approved biosimilars is 36, and it is increasing day by day. Yeah, so this slide is talking about biosimilar uptake and market share in USA. So this graph you can consider, this is a time frame, and this is a growth by volume. So if you talk about last 2 years, from 2019, the launched biosimilar in last 2 years, the average market share is about 65%, and before that it was just about 13%. So infliximab, then filgrastim, so all those are having low market share. And nowadays, because of adoption has been increased in United States, physicians are prescribing more toward biosimilars. It has been seen the momentum. So around more than 50% market share has been reached in last 2 years for biosimilar products. Now, if we talk about biosimilar price at launch versus reference product, so what kind of discount is there in case of biosimilar compared to innovator product? So it has been seen in the USA that a biosimilar uh, is getting introduced at a 3% to 24% price below to the reference product ASP. So the upper part of this graph is showing biosimilar WAC versus reference product ASP. So there are two kind of prices. One is wholesale acquisition cost, and another is ASP, that is average selling price. So WAC is basically a price which is not discounted, and ASP is basically price which are discounted. So if we talk about what will the price of biosimilar compared to reference product, so once it is launched, it is close to around 15 to 37% lower than reference product WAC. So here there's a huge difference between biosimilar as well as biosimilar and generic products. In case of generic, this price is eroding like 80%, and here it is not more than 40% you can consider. Then we have seen there is a huge saving in case of biosimilar in United States. So if you see in the last 2 years, there is a huge saving when biosimilar the number of biosimilar getting increasing and they are getting launched. So this much huge saving is coming from oncology segment. So trastuzumab, bevacizumab, pegfilgrastim. So all are contributing to the saving of biosimilar product. So the savings are coming from biosimilar product because of their discounted price. They are having discounted price. So the cumulative saving is around 9 to 8 billions over the last 5 years. So this data is from last 5 years, and close to 10 billion saving we can consider for the US healthcare system. So this is showing a huge potential in coming year also. Now this is about forecasting. So in the last slide we have seen the trend, like up till 2021 Q2, the cumulative saving close to 10 billion. But in 2025, the cumulative saving from biosimilar is expected to exceed 133 billion, and it is in the range of 85 to 183 billion. So here there is a huge momentum we can see. So the time will say, but yeah, in the trend we can see there is a huge opportunity in the biosimilar. Now if you talk about biosimilar scenario in Europe, we will see centrally approved biosimilar uptake and competition, and saving from biosimilar competition. So you can see this is a graph of approved biosimilars in United States between 2006, where the first biosimilar got approved as a filgrastim, until 2021. So in the last 3 years, you can consider there is a huge number of biosimilar getting approved compared to previous years. So these are basically oncology driven, or the biosimilars which are in the oncology therapeutic segment. So you can consider there is a huge growth in the biosimilar product approval in the Europe also. Now if we talk about biosimilar market share, or you can consider the uptake of biosimilar. So this slide is showing like market share of biosimilars. So the product which are approved in last 2 to 3 years are having huge market share compared to the product which are approved previously. So you can consider from the October 2013 infliximab, the yellow color or yellow to orange color, and there is a, you can consider low market share, but from 2013 you can see the increasing number of biosimilars coming. And if you see in the 2020 bevacizumab, the blue line, uh, close to 60, more than 60% of market share has been converted to biosimilar. So biosimilars which are launched in the past year have reached about 50% of penetration of the originator within less than 1 year, while previous molecule took over 2 years. So this is showing like day by day physicians are having good confidence about prescribing biosimilars. They are looking for the real time data, and because of those promising data with respect to safety and efficacy, physicians are more confident for prescribing biosimilars, and the awareness for the biosimilar are getting increased day by day. And this is the momentum where we can see a huge penetration of biosimilar in the European market also. Now the dynamics of market penetration as well as price discount are like it varies based on different country to countries, like countries regulatory policy, countries reimbursement policy, countries pricing policy. Then in most of the European countries, it's like a tender based business. So those dynamics play a huge role in case of biosimilar market penetration as well as price discount. So you can consider like in all these countries, even with different countries, you can see a difference in market penetration and price discount. So in mature European markets, biosimilar adoption exceed 46% by volume across all molecules. However, this aggregate number mask wide variation in uptake between individual markets and molecules, resulting from differences in policy, utilization of tenders, pricing and reimbursement, and prescriber education. So there are many factors which are playing for the uptake of biosimilar, and sustaining the business in biosimilar is not that much easy compared to generic, because you have to play with all the reimbursement policies, pricing policies, what are the aspects associated with tender business, and what are the competitors pricing policy. So all these will come into picture, and uh, that way it's not easy to sustain in market. But yeah, once you grab the market, once you have higher market penetration with the confidence of physicians and with the awareness of patients, once you see the promising data with respect to clinical trials that your product is very much similar to the brand product, you can get higher market share and you can leverage good business in all these markets. This is long-term view on list price savings from biosimilar competition. So again, the data says in the last uh, 3 to 4 years, there's a huge saving from biosimilar medicines. So this is same like I have told in the previous slides for USA market. This is for EU market, and here it is accounted for around 5.7 billion in saving versus the pre-biosimilar cost of originator by 2020. So this much of saving has been recorded, and this is primarily due to launch of adalimumab, that is the yellow color. So in 2019 or late 18, the adalimumab has been introduced into the European market, and rituximab also introduced. So all these oncology as well as immuno inflammatory products are contributing toward higher saving in European market. Now, if we talk about biosimilar scenario in India, and here we'll talk about history and updated guideline. Just a small part we'll discuss, and then we'll discuss waiver of efficacy studies, interchangeability, and extrapolation of indication. So if we talk about biosimilar scenario in India, so as we have seen in the first slide, biosimilars are termed as a similar biologics. So they are not termed as a biosimilar, but they are termed as a similar biologics. And it's much like a story about a work in progress. So previous to July 2012, there was no rigid guideline. The biologics and biosimilars are regulated on case by case basis using abbreviated pathway for the new drugs, and the review committee on genetic manipulation, that is RCGM, and SADESCO, those are evaluating biologics on case by case basis. But with evolution of biosimilars in different countries like US and Europe, and changing dynamics in the regulatory also, there was a need for a uh, guideline on biosimilar or similar biologic product for India also. So the first guideline on similar biologics framed by SADESCO and DBT which was came into effect from September 2012. So before that, there was no strict guideline, and there were many products which got approval before this guideline came into picture. But since 2012, the guideline was not meeting the global standard. There was a need to update or upgrade these guidelines. And on August 15, 2016, the Indian Regulatory Authority released updated guidance. And this guideline is quite matching with the global standards with Europe and US regulatory agency. And uh, uh, this guidance also addressing waiver of efficacy studies, like in which cases there will be uh, in which cases regulatory agency can give waiver for efficacy studies. So then after, more than 60 biologics got approved, and almost half of them are similar biologics. And on the cost side, if we see, so it's been seen like approximately half of the cost of innovator product, the biosimilar or similar biologics are into the market. Now, if we talk about waiver of efficacy studies, so as I mentioned before, like based on 2012 SEDESCO guideline, nearly 20 biosimilars were approved, like infliximab, etanercept, adalimumab, rituximab, and many more biologics got approved and received approval in the Indian market. However, those approval are with limited clinical studies. So the data on safety and efficacy were very limited. But now with the present guideline, the present guideline suggests that a waiver can be provided to confirmatory efficacy and safety study if there is a high similarity with the reference biologic with respect to structural and functional characteristic, preclinical and PKPD outcomes. So they are basically uh, they're basically highlighting that if the biosimilar company will come with a good data package of good similarity assessment, uh, sophisticated techniques which are used to characterization of biosimilar that has been used, and if they show good matching PKPD profile, in some case they may allow for waiver of safety and efficacy study. However, this is still need to be matured, and still the guideline need to address like in which scenario they can go for the waiver of clinical studies. Like in Europe, MHRA, UK, which is separated from Europe. So MHRA is now open to accept the waiver of efficacy studies if you will show them a good data packages with respect to similarity assessment and analytical techniques, good PK profile and robust process. They may accept efficacy waiver. But yeah, that is a case by case consideration. Now, when we talk about biosimilar, interchangeability is a very key aspect, and this need to be understood by each and every company who is developing biosimilar. Why? Because unlike generic product, which is easily substitutable at pharmacy level as well as physician level, uh, in USA, biosimilar product is not directly interchangeable. So US regulatory authority, US FDA, is assessing interchangeability, and they're giving designation like this product is interchangeable. So once that product got interchangeable, it is expected that product is easily substitutable from pharmacy as well and from physician also. So for that, USFD is asking for switching studies. So apart from normal safety efficacy trials, normal immunogenicity studies, this is additional requirement in United States where they are assessing interchangeability. So the player or the company need to perform additional switching studies, and they need to showcase like with scenario switching of biosimilar and originator product, and there is no change in efficacy or the product is there is no clinically meaningful difference between original product as well as biosimilar. So that is additional requirement in USA, and companies are nowadays trying for that also, because they need to get good market presence. So once they have interchangeable designation, their product will fly. It is expected to fly because it is substitutable with the brand product. So in USA, there are two products which are approved with the designation of biosimilar. One is insulin, Simgli product, and second is Siltezo, that is Humira biosimilar. And many biosimilar companies have submitted supplements for the interchangeable designation for many of their biosimilar products. Now, if we see in the Europe, so Europe does not evaluate interchangeability of biosimilar, and the approval of interchangeability is designated to the member states, because Europe is a bunch of many countries. So there, it has been the power has been delegated to member states to assess the interchangeability. And in case of India, the interchangeability aspects are still not addressed. In India, usually the interchangeability or substitution you can consider is made randomly by the prescriber or pharmacist based on product cost and assumed patient affordability. So here there is a scope of improvement for Indian guidelines, where they address interchangeability and extrapolation of indication kind of approach. Now coming to extrapolation of indication, this is again an interesting topic when we talk about biosimilar. Now if you see like the originator product, so they need to perform trials for all indication. Let's say there are 4 indication, so original product or reference product need to perform trials for all the indication. But there is a guideline in the USA which says if you perform a clinical trial for one indication and show that your mechanism is working, and you show there is a similarity in efficacy and there is no clinical meaningful difference in one indication, then it can be extrapolated to other indication also, provided you provide them in a supplement like high degree of similarity in physicochemical properties, comparable PKPD and immunogenicity profile. You need to prove that your mechanism of action involved in that indication tested and in the extrapolate indication should be same. So based on totality of evidence, without performing a clinical trial for indication, you may get extrapolation of indication. So you need to perform a clinical trial on one indication, and based on justification, which is justification based on mechanism of action which is similar to other indication, you can get extrapolation of indication. And that kind of dialogue is a key regulatory strategy when you are developing biosimilars for different countries. So that is a key part when we are developing a biosimilar for global market. So as I told, like this is one case study for Humira biosimilar in United States. So there are almost 6 biosimilar. Currently there are maybe 7 or 8 biosimilar, but in this case study you can see the yellow part. So that indication that they have performed for clinical trial, and raised the right sign you can see, those are extrapolation of indication. The companies are not required to perform clinical trial for this indication. Why? Because they have proved that the mechanism of action who are responsible for efficacy of this indication are same as of the indication which are studied. So that way in the US, this is one key provision for extrapolation of indication. Now in the same case, rituximab, there are 2 indications. So rituximab is indicated for non-Hodgkin lymphoma and also for rheumatoid arthritis. Now for both these disease, there is a different mechanism of action responsible. So one cannot go like I have performed a clinical study on this part and I need biobehavior or I need extrapolation of indication on this part also. So where there is a different mechanism of action responsible for different indication, one has to perform additional trials, right? So now I will summarize like what are the key points to consider while going global, and then we can consider all the question answers. So this is a key part of my presentation where I'm discussing what are the key steps or key points one company has to consider. So first and foremost is market size and forecast. So whenever we strategize like we need to go for this country or we need to develop biosimilar for this country, first of all we see like what are the market size for that product in that region. So country-wise sell, volume and growth trends. It's historical data like what is the growth in the past years, and forecasting activity like in the coming year what will be the size of this product in that particular region. So after assessing the market and forecast, we'll come to the therapeutic competitive landscape. So this is very much required when we are forecasting also. This is required to assess advantage of biosimilar product in comparison to already available treatment. So we need to assess like there is one product available in market. Now if we follow that product in the future, the another company will come up with different product which is more efficacious or which is more advantageous to already existing product. So the product which you are following, it may not be that much having good market size, and if you follow that product you may end up with losing the business, right? So that assessment is very much required when you are developing biosimilar, because it's not a kind of generic development we are doing. For biosimilar it is required a huge cost and timeline, so you need to assess future also. Then we assess launch scenario for like whichever market we want to go, what are the patent and exclusivity scenarios. So when the patents are getting expired, whether we can bypass that patent or not, an exclusivity scenario. So all this will be helpful in predicting launch scenario. And so based on launch scenario, we can set up our goals like within which time frame we can launch this product and when we can start. Then fourth is market competition. Market competition basically how many biosimilar players are developing that product which we are developing. So that is a direct competition, and that competition we need to assess like if there is a if there are two players or three players, what will be our market share and what will be our business case down the line when we will launch our product into the market. So that assessment is a key which needs to be done. Fifth is technical capabilities. So of course, for the biosimilar company, it is very much required to have highly skilled manpower and high-end characterization techniques, because these are the base of biosimilar development. And the company which are the companies who are having more of this kind of resource, they can leverage their expertise and they can win the business. Then process development, as I told, like process are patent protected, and one has to bypass those processes. So efficient processes which can provide better yield. You need to focus on yield also. Only developing a product is not uh, uh, you know profitable, but yield part also need to consider, and that's why a low cost of product we need to develop. Quality plus low cost of product with better yield. Seventh part is clinical and regulatory affairs. So when we are going global, your clinical team or clinical strategy and regulatory strategy should be aligned with different regulatory agencies. You need to start a dialogue with different regulatory agencies with respect to your clinical protocols, your clinical study framework, and regulatory pathways, right? And if you are asking for waiver studies, you need to prepare with all the justification. So that clinical and regulatory affair team should be very strategic. Then manufacturing capabilities. So companies need to have robust and easily scalable processes to avoid any surprises in the manufacturing. High volume capacity which can serve global requirement. So if we are going global, you need to assess what are the capacity which company needs to require, how many bioreactors, what size of bioreactor requires. So all those assessment is required prior to we go into the market. And of course all this would require analysis of budget part also. Then coming to marketing structure, once we have approval, it is not like we have in the business. You have to have strong in-house marketing team with regional sales director, sales representatives. Here it is not limited to sales representative. You need to have account managers, reimbursement specialists, medical science license, group purchasing directors, payer directors. So all these will help in creating awareness for biosimilars, educating physicians and nurses for biosimilar product, right? Uh, framing the reimbursement policy also. So all these would required when you are into the market with biosimilar product. So it's a huge in-house team that is required, and with the skilled team we can create a good reputation in market with good market share. And last but not least, patient assistant program. So these has to be into the market like brand. So you have to promote it like a brand, and you have to provide patient assistant program. You need to provide financial assistance to help patient access the medication for little or no cost. So that's that kind of hub services you can consider a key part of marketing strategy, and you have to have that website to promote your product. Thanks a lot.
Okay. Thank you, Kishan. It was a wonderful talk.
Thank you.
Basic understanding related to the biosimilars across the globe, particularly three major markets: US, EMA, and the India. Of course, we missed Japan. However, I'm sure that in the coming time probably we'll cover Japan also. So, if we have any questions from the audience to Mr. Kishan, 2 to 3 questions. Anyone?
So hi Kishan, I would like to ask a question.
Yes, please go ahead.
Sure.
A very nice PPT. I'm really impressed. Very good data set.
Thanks a lot.
So basically you were talking about waivers, and like Indian government can give waivers, right, while application and approval process. And you are saying that PK/PD study will still need to be made, right? Or it will be a waiver even for PK/PD study?
No, so it's like that. Basically, first of all, let me tell, it's a case by case basis. Of course, there is a guidance, but in US also it's a case by case basis. But in US, the scenario is like you have to have at least one clinical trial. Without that, you cannot get approval for biosimilar. In Europe, it's a case by case basis. Let's say in UK, they are now open with a waiver of safety, waiver of efficacy trials. So there is a scope. Now in Indian case, it still need to be matured. Like in the guideline, it has been written, but yeah, one has, so PKPD has to be performed. Phase 1 trial has to be performed. With respect to phase 3 trial, that is totally based on totality of evidence. Like what is your data package with respect to similarity assessment with innovator product, because innovator product is already in market since many years in Indian market, right? So Indian government is considering 4 years as a timeline. Like after 4 years, it's not considered as a new drug, right? But in case of biosimilars, clinical trial is assessed by case basis. So once you have phase 1 data and similarity package with respect to sophisticated analytical techniques, bioassay, then you can submit a waiver request for Indian SADESCO, and then
Oh,
case by case they consider. So
okay,
that
okay,
still under process, and it's not matured enough as
okay.
like in US.
Okay, but still in India, we have like 20 of them approved with a limited clinical trial data.
Yes, 20 and
Okay.
many, 20 and
Okay.
many approved now though.
Okay.
So yeah,
Okay.
but
Okay.
that is a process Indian government is also taking care, and it's as
Okay.
I mentioned, it's a uh, process is streamlined now. But yeah.
Okay. So just another question from business development perspective, that you have shown that like there is a severe, not severe, there's a huge savings on the government part through introduction of biosimilar and all.
Right.
But from the part of investors, right? So if you are the first or second biosimilar maker in the market, then you will be really profitable. But whenever you are working for any specific biosimilar, you already know probably that how many players are already working
Mm,
and at which stage they are, right?
mm, right.
So what is your like decision making at that time? That how do you take decision that whether we should go for working to develop this biosimilar or not?
Right. So coming towards your first approach, that is how many players are there in market. So as you rightly mentioned, within one or two players, there is a good business case that we can consider. But after three or four biosimilar, if you are entering to market, it is somewhat difficult. I'm not telling like it's gone case, but
Mhm,
there are a few case study also on that. Like if you are in a market with good data package, because you need to convince physician also. Even after approval of biosimilarity, you have to go to the physicians. Your how strong your distribution channel is there, and how strong your marketing team is there. So that is also one key aspect, because it's not straightforward reimbursement policy, and in United States payers are playing major role. So the player who are having good reach in those areas are getting leverage for market share. Let's say talking about Kasir Bioscience where I'm working. So in US they are preferring US made product. So if you're plant or if you're manufacturing in USA, so that is preferable for all these reimbursement directors or policymakers, payers as well. So that is also one factor which play a role.
okay. And with respect to other markets like South American, African market, they
So
won't afford.
emerging,
But yes.
yeah, yeah. So emerging markets, biosimilars are not very much matured now, as of now,
Okay. Okay.
and they are still following different practices. It is not that much streamlined. They are taking products from regulated markets. Like if any company who is having US FDA approved product, then they are directly registering that product into those market. And of course, there is no higher regulatory agency than US FDA. So those products are considered as a approved product. Of course, they have their
Uh,
own checks with respect to stability data, like region wise. On higher temperature, maybe they require stability
yes.
data, but that is minor work that companies need to do.
But I mean, usually what happens is that people go for ROW first, then for US and EU,
So that was
right?
yeah, that is scenario a few years back, where Alvogen has that kind of model. And
Okay.
because in Europe, the entry of biosimilar was very uh, previous to in US. So
Yes.
since 2006, they have first biosimilar, and
Okay.
after 10 years in US, there is first product.
Yeah.
So because EMA, European Medical Medicine Agency, was very much mature, and they know about biosimilar regulatory regulation and what are the data required since long
Mm-hmm. Mm-hmm.
back then. US, in US it was in 2010 they came up with BPCI Act and all regulation they have framed.
Yeah.
So that is why the company had approached like let's go to the EMA. They have
Okay.
good framework of
Exactly.
regulation, and then we'll come back to US.
Alright.
But
Okay.
now it is not like if company wants to start with US, they can start because it is very much matured now in US also.
Okay. Okay. Thank you very much.
Yeah.
Okay, Kishan, I don't know whether that question is valid or not, but I'm just curious. For example, whenever an innovator company wants to do a efficacy study, and if they are planning to launch a product in US, they will consider, the patients from the US market. So when we are developing the biosimilar products, the bioequivalence normally US and EMA consider from Indian the healthy volunteers. But when it comes to the clinical evaluation, even the single indication for a biosimilar, any subject from any part of the globe would be acceptable to a US or Europe. I don't know that clarity.
Yeah, so it's like biosimilar companies can have population from different region. Let's say if I'm developing my biosimilar for US FDA, right, and the innovator product is in USA, and I'm following that product as a biosimilar. So as a biosimilar company, it is possible, and even US FDA can also accept a patient population from different countries. But majority Indian companies are taking patient from India because it is accessible, cost effective. But of course, it is not like we can rely on 100% India population. It is case by case basis, but US FDA tells like you need to have at least 20 to 30% US population also. So it is not like 100% Indian population would suffice.
Okay.
Right? So
Unlike the bioequivalence for a small molecules where it is not any,
no
it was
clinical
never,
trial would be required.
yes, no clinical trials.
Yeah, it is only based on bioequivalence. So there
doesn't
it
matter
is.
volunteer from where you pick up. So
Yeah,
here.
and it is on healthy volunteer. If you talk about generic medicine, we do it on healthy volunteer. Here if you talk about phase 1, it's on healthy volunteer, but if we talk about on phase 3, it's on patient population. So patient population you can take it from India, Europe, and US majorly.
Okay.
So that protocol the clinical team would discuss with the regulatory agency US FDA, and yes, in many cases they accept that kind of blend of population. Let's say 70% Indian, 10% European, 20% USA, because for biosimilar they just need to prove their mechanism of action working, and they just need to show non-superiority. So it's efficacious. That is only requirement. So that kind of blend is possible.
Correct. Okay. And the last question, I don't know how important that question is, but it is my observation that when the innovator company launch their biological products, over a period of time, there the manufacturing process with respect to the impurities goes on extremely low levels. By the time now the time comes for the launch of the generics, maybe it may be related to the expiry of the patent, or the expiry of the exclusivities. The impurity level in the drug product of that biological product is extremely the low levels. Here the question is how this biosimilar company, because it's like I'll say unnecessary work done. You are trying to have an extremely pure compound, because we are looking, as you mentioned, yield and the cost. Both
Yes.
are now that is. So how the biosimilar company are overcoming this issue?
Yeah, so in case of biologic product, innovator files a patent like anything. So there are a thousand of patents for a single product. So those patents are on process, impurities. Those can be on different indication, different dosing regimen. So there are thousand of patents for impurity process. Like, so now as a biosimilar company, if those patents are hurdlesome, then you need to tackle. So biosimilar companies need to change their process accordingly to remove those impurities and to fine tune the process according to the development, right? So that way it's a challenging process for biosimilar.
Okay.
I hope I have answered your question.
Yes.
If not, you can clarify once again.
I would just add one statement to it. The QBD that like I was discussing probably in the last slide, but I was not had enough time to discuss it.
Yeah.
That is the basic approach which regulators also say. So that is a really very useful thing. That is a
Yes,
very
yes.
robust way of determining your process impurities, process parameters, and everything. So, I mean,
So
having that in place clarifies most of the things.
that's true. And in addition to that, in case of biosimilar, you need to have good scalable process also, which is robust process. Because let's say if you have made a product on 5 liter, 10 liter, or 50 liter, but then what if you cannot scalable, and if your impurity levels are getting higher in case of 1000 liter or 500 liter, then your commercial plan would be shaky. Like you need to have approval for your commercial batch, and then if you will scale up and if your impurity level will go on increasing, then it would be difficult to justify for USFD also.
Okay. And the last question, it's my observation that, our Indian pharma companies are going little bit slow to the US market. What could be the reason in your opinion? Is it, this uncertainty related to the patent scenario, or the extreme complexity in the, the development of the biosimilar? What is your opinion about that?
Yeah. So let me share 2 to 3 perspective on this question. So first is why they are not going to US, right? That's what you are
Right.
your first question. So they are starting from Europe. Why? Because Europe is having biosimilar products since 2006, and they feel like getting approval in Europe is quite easier than US FDA, because US FDA has evolved with the law with 2010. So they have first product in United States in 2015, after 10 or 11 years of first approval in EMA. So what Indian companies are thinking, because it's a huge cost associated, first thing. Second is timeline is very high, 6 to 10 years you can consider for developing one biosimilar. Third part, there are huge patent protection. So in US, patent laws are different. In Europe, patent laws are different. So in many cases, it is happening like in US, the patent are getting expired later on. In case of etanercept, one example I would like to say, etanercept, Sandoz has approval in US since 2 to 3 years back, right? But the case or lawsuit has now settled, not settled, but Sandoz has lost the lawsuit. And because of that decision, Sandoz is barred to launch their product since 2018, 2028 or 2029. The same product is launched in EMA, Europe, because of there is no such patent protection. Now coming to patent protection in US, the patent laws are pro innovator. Like the innovators are having many patent protection on different indication, different dosing regimen, right? But in Europe, it's bit challenging to get those patent, and you can easily invalid those patent if there. So that is those are the key 2 to 3 reason why Indian companies are going towards European approval and then coming back to US. And of course, cost part is a huge, because Indian companies are not innovator companies; those are generic companies. So they had to pump a lot of money on their facility capabilities, manpower. So that is also a cost which is a risky. So biosimilar is in one line, high risk, high profit business, but high profit is not guaranteed. Uh, you need to uh, you need to work on your marketing strategy also. So that is why Indian players are more towards European part, and then coming back to US.
Okay, got it. All right, so we have reached to the end of this webinar. So once again, on behalf of NIPER-Ahmedabad, I would like to thank our speaker, Dr. Roshan Goswami and Mr. Kishan. It was really a wonderful talk and we enjoyed it. And at the same time, we also would like to thank our all the participants who have, joined this webinar. And with this, we are ending this webinar, and we will reconnect, with a new element of the biosimilar development, maybe like, the patent perspective, or maybe, the downstream processing or the upstream processing. We are going towards more specialized topic. So once again, thank you all for, making this webinar a very successful one. Thank you all.
Thank you.
Yeah. Thank you very much.
Thank you very much.